So, in the last class we saw a demo where we saw a example of a bioinformatics tool that was capable of efficiently analyzing large number of SARS-CoV-2 genome sequences. In this class, I want to go over the basics of molecular biology and high throughput sequencing in a very minimalistic view. So, the goal here is that for students with engineering backgrounds who lack experience with molecular biology or high throughput sequencing, I want to give them a brief overview of the biological concepts and the sequencing concepts. This will help us later because when we are motivating different problems and different biological requirements, we will be referring back to these biological applications, and then this knowledge will be useful there. So, as part of the introduction, I want to go over the basics of molecular biology and high throughput sequencing. So, first I want to tell what DNA is. So, a DNA molecule is typically a double stranded molecule, meaning that it consists of two chains. These two chains are present in a double helix like structure, as you can see on the right. Each chain is composed of 4 types of nucleotides: adenine, thymine, cytosine and guanine. Because this molecule is formed of only 4 types of nucleotides, I can even simplify this picture and I can represent the DNA as a sequence of A, T, C and Gs. Now, so because DNA is forming a double helix like structure, what I want to emphasize here is that if one of the strand has A character in it, then the opposite strand will have T character in it and vice versa. So, A and T are complementary bases. Similarly, if you have C on one strand, then you will always have G on the other strand and vice versa. So, you can think of in some sense that if you know the first strand, then you also know the second strand, because I just need to complement these bases and I can know the second strand as well. And this redundancy is maintained in DNA because it allows for efficient copying of the DNA when the cell splits into 2 cells. Also, these complementary bases exist because the force of attraction between complementary bases also helps to hold the double helix like structure together. Now, the complete DNA sequence of A, T, C, G and so on forms a complete chromosome, and the complete chromosome, so these chromosomes reside inside the nucleus of the cell. The set of chromosomes, the entire set of chromosomes is called the genome. For example, in a human we have 23 pairs of chromosomes from 1 up till 22, and then sex chromosomes X and Y. So, one copy of the chromosome would be inherited from father and one copy of the chromosome would be inherited from mother. Now, coming to the central dogma of molecular biology, we also know that DNA is transcribed into RNA and the RNA is translated into protein structures. So, for example, so the goal here is to emphasize that the DNA contains the instructions for the downstream parts. So, proteins are going to be responsible for large variety of functions that are carried out in our body. For example, you must have heard of hemoglobin, antibodies. These are all proteins that are necessary for the body to function, and DNA acts like a program that specifies instructions to assemble proteins. Similar to how DNA was composed of 4 types of nucleotides, all human proteins are chains of 20 amino acids. So, when we are writing DNA sequences, we will always use 4 types of characters A, T, C and Gs. For protein sequences, we will be using 20 distinct characters corresponding to the 20 amino acids. Now, going into the more details of this, how this DNA gets transcribed to RNA and then RNA converting into proteins. So we have to go and look at the entire DNA sequence. What the first term that is important again is the gene. So genes are the intervals on the chromosome that are transcribed into functional RNA or proteins. So there will be segments on the chromosome, for example here shown gene 1 and similarly gene 2. These are intervals on the chromosome that are transcribed into RNA. The remaining regions of the chromosome will not be transcribed to RNA. So once the transcription happens, so suppose this gene 1 gets transcribed into pre-mRNA. So this is the first copy of the RNA. Here the DNA sequence will be converted into the mRNA product. Within the gene there will be segments known as introns and exons, and what will happen in the mRNA product is the splicing, where the introns will be discarded and only the exons will be kept in the mature mRNA transcripts. So only these grey regions represented as exons will be coming into the mRNA transcripts. Subsequently these mRNA transcripts will be translated into protein sequences. So at the translation step, consecutive 3 nucleotides will be converted into a single amino acid. So this is how the conversion of DNA to RNA and RNA to proteins happens. Now you may wonder, how come each cell of our body has the same copy of DNA, but still we see a variety of cells. For example, we have brain cells, we have muscle cells, we have blood cells. They all look different from each other and they perform different functions. So how come these cells, despite having the same instruction to code the proteins, they still work differently? The answer lies in the regulatory mechanism, because there is a regulatory mechanism in the body which can turn some of the genes on and off. So in each cell, depending on the requirements, the genes can be turned on and off. For example, blood cell may need to manufacture hemoglobin, the immune cells may need to manufacture antibody proteins and so on. So the way this regulatory mechanism works is as following. There are proteins, special proteins known as transcription factors, which can interact back with the DNA sequence, and depending on, so there are specific regions in the genome where the interaction can happen. So these regions can be known as promoter sequences or enhancer sequences or silencer sequences, and these transcription factor proteins can bind to the DNA, and they have the capability to, this interaction has the capability to turn the gene on and off. So they can regulate a particular gene depending on this interaction. So this serves as a regulatory mechanism in which the transcription level of each gene can be manipulated in different cells. So these particular regions where the transcription factors proteins bind are known as transcription factor binding sites, abbreviated as TFBS. And this machinery runs for different genes. So for example, the same thing will happen at gene 2, gene 3 and so on. And on a human genome has about 20,000 genes, and that is why with this regulatory mechanism now you can imagine how blood cells and all different cell types function differently. Now it's another point I want to emphasize is that the genome sequences that we have, they are almost identical, but on an average they are 99.9% identical, and that 1.1%, 0.1% difference makes us unique. So for example, my eye color or other aspects like height are governed by such difference, can be governed by such 0.1 differences within the genomes. So the mutations in any human being are of several types. For example, you can quantify these mutations with respect to a particular reference. So for a human genome, we have a human genome reference, which can be thought of as a consensus genome of the species. And the way I would quantify the mutations in my genome would be by matching my genome against that reference and quantifying where the substitutions or where the insertions or where the deletions are happening. So, the top could be the reference sequence and the bottom could be my genome sequence. And therefore, the mutations can happen in various forms of substitutions, insertions and deletions. The substitution mutations are the most prominent among all types of mutations. Now, the goal of high throughput sequencing technologies is to study DNA and RNA molecules. The picture that I showed you for the central dogma of DNA converting into RNA and RNA converting into proteins, the high throughput sequencing technologies enable us to get a view of that process. For example, if I want to know the DNA sequence, I can use the whole genome sequencing technology, which will go and read the DNA sequence in the cell. Typically, the way whole genome sequencing works is that it will not read the entire genome sequence end to end. It will fragment the DNA, and then we will see the fragments of the DNA converted into strings, and the output of the sequencing instrument will be a large set of strings. So, these are represented as black line segments here. Second, if I do not want to read the entire DNA sequence, but I am only interested in, let us say, a certain gene or a particular segment of the genome, then another technology that I can use is targeted sequencing, where I can use the primer sequences to target specific regions and only sequence within that specific region of interest. Similarly, if I want to study RNA sequences and I want to quantify the different type of RNA sequences in a cell, I can go and look RNA sequencing, where RNA sequencing will go ahead and sequence the RNA molecules inside the cells. So, here you will see the fragments of sequences coming from the transcripts, and this will allow you to quantify the RNA molecules in the cells. Finally, I also mentioned that there is a regulatory mechanism in which transcription factor proteins can bind to the DNA and regulate the transcription level of a gene. For this, if I suppose I am only interested in sequencing the areas of the genome where these transcription factors are binding, then I can use the technology known as chip sequencing, where only the regions where these transcription factors are binding will be sequenced and the remaining regions of the genome will not be sequenced. There are many other technologies which are not being covered here, but the message here is that there are high throughput sequencing technologies which can generate a lot of data, and they allow us to understand the molecular mechanism by reading the DNA, RNA or protein sequences. One can imagine that there are a lot of applications of high throughput sequencing. For example, in agriculture, I may want to sequence crop plants and understand the properties of crops that will make them protect them against heat resistance, or I may want to do environmental sequencing to track or to surveillance of infectious diseases, precision medicine, evolution of species, forensics and even genetic counseling or newborn screening. So, there are many application areas where one would use biological sequence analysis, and that is where the importance of algorithms comes into the picture, because all these sectors, when they are using high throughput sequencing, they will be generating a lot of data, and for the analysis of that data will require efficient algorithms that we will be studying in this course. So, in the human genetics also, the application of genome sequences is to sequence people. So, for example, the first human genome sequence came in 2001, and now because the sequencing technology has become so ubiquitous, we can even sequence large populations in different countries. So, an example is the Genome India project, which is sequencing 20,000 individuals of India. Similarly, during pandemic, you may have encountered genome sequencing labs at the airports where they would take the swabs and sequence the pathogens to know the genome sequence and track various SARS-CoV-2 variants. I would also encourage you to look at this link below and see various milestones in genomics, because it will give you a historical context of what has happened in the last 30 years and what milestones have been achieved in genomics field. So coming to the big data. So where is big data in this high throughput sequencing? Well, the big data comes into the picture because if we look at the last 20 years, the cost of sequencing has dropped by 6 orders of magnitude. Sequencing the first human genome was very expensive, it costed billions of dollars, but now sequencing a human genome is a routine task. In 2001 there would be 1 or 2 central facilities in the whole country which were capable of sequencing. Now each lab who is looking at molecular biology would have their own sequencing instrument. Therefore, the data generation is happening at massive scale and the size of public databases is growing exponentially. You also see a comparison of this drop in sequencing cost with the Moore's law. This is to show that we cannot just rely on the computing power, because each year computers are also getting faster, but the rate of at which these sequencing technologies are becoming ubiquitous and cheaper, that has outpaced Moore's law, and that is where the algorithms all have to also catch up and process this data faster, because we cannot just rely on the computing power. You can also go ahead and sequence your own genome. So there are commercial vendors in which will take your saliva or blood samples and that can give you genome sequence of yours. The last point in this class that I want to emphasize is that the sequencing technologies have been evolving as well. So if you look at 2008, this is the Illumina sequencing instrument. This instrument was capable of producing 1.6 billion nucleotides per day when you are doing a whole genome sequencing experiment. You can see how the throughput has been advancing year by year. So if we continue to go and see 2017, the throughput has increased to 1 to 3 trillion nucleotides per day, and one cannot utilize naive algorithms to process this amount of data. Therefore, the motivation of this course is to build algorithms that can utilize and process this data and make interesting biological insights so that one can enable scientific discoveries and help the applications involved. Thank you.